Hello, are we live?
Hello, are we live?
Oh, let me mute this.
Hello, are we live?
Oh, let me mute this.
Hello, are we live? Oh, let me mute.
Sorry about that. How is everybody? Where's everyone joining from today? Let's do a little competition while waiting for everyone to join. See who's from the furthest away place. Hi, good evening. Kenya, nice. Caribbean, Jordan, Dubai, Barnsley, represent. Nice. Singapore, that's pretty good. Australia, oh, I think you might be winning so far. How is everybody coping with lockdowns or whatever you've got in your countries at the moment? If you're joining from the UK, then good news is we've only got, is it like 5 more days now of lockdown, and then we're free. I'm dialing in from London today. So yeah, in the UK. And yeah, for those of you not in the UK, we've been on a month lockdown. So we're coming out of that again next week. Although I think it's still going to be we can't do that much stuff. Okay, so yeah, thanks for joining this evening. This YouTube revision class from SnapRevise. We're going to go through DNA and RNA. So this comes up in year 12 for all the specs, at least all the UK specs. And it's a really important part of biology. So that's why I picked this topic as one to go through. So no matter what exam board you're doing, you will need to know about DNA and RNA. So it's useful stuff. So without further ado, let's jump right in. I'm going to share my screen and we'll go through it. Okay. Oh, one sec. Perfect. So yeah, the class today will be on DNA and RNA. A little bit about me before we begin. My name's Alex. I'm the head of biology at SnapRevise. I've been teaching in London and online and tutoring for the last, it's probably actually closer to 5 years now. But yeah. And yeah, so we're going to go through DNA, as I mentioned. So a little info on what we're giving away today. We've got some prizes up for grabs, so very exciting. You can win some things by attending this class. So not only do you learn, you also get the chance for a prize. So we're giving away 1 free account at the end of this session. If you stick around to the end, we'll explain how you can do. Oh, sorry, this one you can enter straight away. All you have to do is follow the SnapRevise channel on Instagram, share a picture of the class, and then just tag at SnapRevise, and we'll pick one of you as the winner, and you'll get a free account. So that's really cool. Everybody will get a coupon code at the end of this class. So this is a discount code to use. So you have a chance of winning, but everybody will get a discount code. So yeah, prizes for all. So the structure of these classes, this is what we do in one of our packages is called the Ultimate Package with SnapRevise, and we do these web classes online weekly. They're normally just for people who are subscribing to SnapRevise, and we do these ones in addition. And the way we run these sessions, at least for biology, is we start with a learning pyramid of topics that we covered in the last session. So for example, the stuff today, this will be things that we learned about the week before, or maybe like the week before that, in the last couple of weeks, just to reinforce what we've gone through. So just in case you were wondering why it seemed a little bit random and it wasn't necessarily relating directly to DNA, that's why this first bit is a little bit different. But it's going to be good revision for all of you. So first question is, what is the induced fit model? So we're looking at enzymes here. Is chemistry next week? I believe so, yes. We change subject each week. So the way we run these, we'd like ideally for you guys to be answering. So when I throw a question out, it's for everybody to answer. The more you engage with it, the more you'll get out of the session. Nice. P-Beans. I like the name. Yes, exactly. So when the active site is changing shape to fit. So Adina, be careful. It's not the same as the lock and key model. It's like it's the improved version of that. So I'm not sure if you were saying lock and key to say it was like different to that, but yeah, just to make sure that it is different, that you're aware it's different. So the lock and key was the first one that scientists proposed. Then they realized that actually proteins aren't rigid. They're very flexible and they can move. So the induced fit one was the improved model. So what can we say? Active site changes shape to fit substrate. Shape to fit the substrate, which was pretty much what all of you guys were saying. So that's really good. Nice. Next question. In which protein structure would we find ionic bonds or ionic bonding? Nice. Yes. Tertiary structure. Perfect. Next up, what are biological catalysts? Oh, I've spelled tertiary wrong. Missed an I. Yeah? So you could give, some people are giving the short answer, some people are explaining what it is. So yeah, biological catalysts are enzymes. And what's a catalyst? Something that speeds up a reaction without getting used up. Getting used up. Perfect. So next question. This is related to the topic today. Oh yeah. Yeah, lots of nice answers. Well done, guys. What is the role of DNA? How would you summarize that in a one sentence answer? Yeah. Yeah, so genetic information is definitely to do with that. So we can say, how we summarize that? Contains the genome, maybe? Genome or genetic information was good as well. Genetic info. What is this used for? And some of you have mentioned it. Yeah, exactly. It codes for proteins. For proteins. So proteins are arguably the most important substance we need to make in our cells, and the DNA has the instructions to do that. Nice. Next, what is a hydrolysis/condensation reaction, it should say, really? Again, so DNA and RNA, they come into the biological molecules topic, which is a big topic you do at the start of AS. And then these hydrolysis and condensation reactions, they come up all the time in this biological molecules topic. So you need to have them like super drilled in and be able to roll them off. Exactly. Nice. So condensation, we're making bonds. And we're removing water. A hydrolysis, we're breaking bonds. Breaking bonds through the addition of water. And we can kind of, like if you ever get them muddled up and confused, just remember, like condensation, when you like breathe on a mirror and you form the condensation, or on a car window, you're like creating the water. So that's the condensation. So when you're creating water, that's a condensation reaction. And another way to remember the hydrolysis is if you split it up, the actual word hydrolysis, the hydro means, or obviously water, lysis means to break apart. So where else would we come across this word lysis in biology? It does crop up in a few other places, but it means, yeah, breaking apart. Yeah, nice. Yeah, photolysis, very good. Glycolysis, yeah, perfect. And in osmosis, we have the, yes, plasmolysis, exactly. And the cells can burst, yeah. Perfect. So yeah, this word lysis, you see it in quite a few places. Okay, next thing before we move on, can you tell me anything about DNA already? Well, I'm sure you can. What can you tell me about DNA? Yes, like that, double-stranded. I'm just going to put DS. Double helix. Yeah, this is going to be like unintelligible shorthand writing, but we'll go through these anyway. Wrapped around histones, yes, like it. Polynucleotide, very good. Deoxyribose, yes, that's the sugar. Phosphate groups made from nucleotides, semi-conservatives, anti-parallel, yes, this is all good stuff and all correct. Oh, give me the bases. Nice. Perfect. So you guys clearly have a pretty good understanding. So this is going to be like a revision session for, seems like for most of you, which is great. So this is what we're trying to cover in this lesson. This is the objectives. We'll have to see how we do for time, because we normally have a whole hour for the lessons, but today, the last 10 minutes, I'm just going to be talking to you guys about the company and showing you our website. So we might not have time to do everything, but I think if we work quickly, yeah, there's a chance we will. Anyway, so we're going to look at the differences between DNA and RNA. We're going to look at the structure of a nucleotide, and we're going to look at how we make polynucleotides from those. And then we'll look at the bonding between complementary base pairs. So these are the spec points. These lessons are they're catered to all UK exam boards. So it doesn't matter which board you're sitting. These lessons will be beneficial for that. So if you want to screenshot your specs, you can refer back to it. Please feel free as I flick through it. Now, this is the OCR ones. You can obviously get these online as well. You can download your specifications, and I would recommend having a printed copy of your spec to refer to from time to time when you need to see what exactly you need to know. So part one, we're going to look at nucleotides and polynucleotides. So what can you guys tell me about the monomers of nucleic acids? And maybe a secondary question is how would you define a monomer versus a polymer? So we should probably recap those as well. Nice, yeah, so the monomers of nucleic acids are nucleotides. Very good. And yeah, mono is a single repeating unit. Or a single, yeah, I mean, that's fine. And then polymer is a chain of these. So again, these words crop up a lot during the biological molecules topic. We have it in carbohydrates with monosaccharides and polysaccharides. We have it in nucleic acids, like we've got here, and we've also got it in proteins when we have amino acids are the monomers, and a polypeptide chain is our polymer. We don't get it in lipids. People often forget this. The structure of a lipid is what do we call this molecule? Looks a bit like a squid or an octopus that's lost some legs. It is a macromolecule, very good. And specifically, this one is actually a triglyceride. A phospholipid looks similar, but it's only got 2 legs and it's got a phosphate group instead. So that's a phospholipid. We find those in membranes. The main one for energy storage, this is a triglyceride. And lipids are different from the other biological molecules because they don't form polymers. So you don't get a long chain of triglycerides all joined together. They're separate individual molecules. So they don't polymerize. They're the odd ones out in that regard. And it's worth, yeah, a lot of people forget that, and they think that they form polymers. So I thought I'd just point that out, even though we're not specifically talking about lipids today. Okay, next question. What is the polymer of a nucleic acid? So there's a few different answers you could say here. Why can't lipids form polymers? Because they don't have groups that are available to bond. So for example, in these ones, we bond two OH groups together, and that forms the continuous backbone via phosphodiester bonds. And the triglycerides don't have an available space to form bonds. It's also why they don't have any OHs. That's also why they're insoluble in water. Sorry, I went off on a bit of a tangent. That was just answering someone's question, which is now long gone because you guys are all answering me for the next question. Yes, correct. DNA and RNA are the two polynucleotides that you'll be familiar with. So you could also say polynucleotide for this answer. So this encompasses both DNA and RNA. So we've got two things here. Hopefully you guys can tell me which one is DNA and which one is RNA, and how can you tell? Yeah. So DNA is double-stranded, and RNA is single-stranded. I'm just going to write SS for that. Cool. So this is obviously our RNA, and this is our DNA. And we'll look at some other differences between those two later on as well. So let's look more closely at a nucleotide, aka the monomer of nucleic acids. So nucleotides all have a common structure. They're made up of three things. We've got our phosphate. Just going to connect the arrows rather than writing them out again. We've got our pentose sugar, and we've got our organic base. So the phosphate, what can we say the phosphate does, or how does it help with forming DNA? Yeah, it forms part of the backbone, the sugar phosphate backbone. Exactly. Nice. So it bonds with the pentose sugar. To form backbone, or sugar phosphate backbone. Let's write the whole thing out. And some of you, backbone, some of you told me the bonds already. So yeah, those are, sorry, can you hear that squeaking noise? Sometimes this bottle, I should have just got a glass. I wasn't really thinking. It makes a squeaky noise sometimes. I was just doing it. Don't know if you could hear it. The bonds that do that are the phosphodiester bonds. Phosphodiester bonding. So the pentose sugar, what can you tell me about this? They're obviously also involved with the sugar phosphate backbone, because we mentioned it here. But what else could you tell me? Yes, Marion, nice. Yeah, ribose and deoxyribose. And where would we find those? Yeah, ribose equals RNA, deoxyribose equals DNA. So this is actually where they get their names from. So RNA stands for ribonucleic acid, whereas DNA stands for deoxyribonucleic acid. So that's actually how they're named. You could probably work out the structure between ribose and deoxyribose. They're quite similar. Deoxyribose has got one less oxygen atom than ribose. Kind of self-explanatory in the name. Those are both examples of pentose sugars. So what do we mean by a pentose sugar? What is that? Yes. Nice, good. I was wondering if anyone was going to say it's a five-sided sugar. Oh, Eleanor, you've fallen for the trap. Yeah, just wanted to point out that a pentose sugar, so pent, like the pentagon, means five, and pentose sugar, this doesn't mean a five-sided shape. It means it's got 5 carbons in it. So whenever we talk about these names, so OSE is the ending for a sugar. So anytime you see a molecule that ends in OSE, you basically know it's a carbohydrate. Just like every time you see one that ends in ASE, you know that that's an enzyme. So we can use this to work out what we're looking at. If you get something that you've never heard of, a big clue, if it ends in OSE, it's a carbohydrate. So it might help you in a test where they give you something that you haven't come across before. Pent means 5. The other options that we come across are like hexose sugars, like glucose, which is 6 carbons, or triose phosphate, which is a 3-carbon sugar. That is referring to the carbon atoms, the number of carbon atoms. It does not refer to the shape. So a pentose sugar could have a different shape to that. And in fact, in the actual shape of the pentose sugar, one of those things isn't a carbon anyway. Oh, sorry, I've drawn it in the wrong place. But one of them is an oxygen. So it's unrelated to its shape. Just wanted to make that clear. Glucose is a hexose sugar. Exactly. It's got 6 carbons. Its formula is C6H12O6. So yeah, hexose. All right, what can you tell me about the organic bases then? A, T, G, and C, and in RNA, U instead of T. RNA. Can you guys tell me what these stand for? Because I was quite shocked by this, actually. I read an examiner report, I think it was from an OCR exam, and they'd asked the students were required to actually give the full names for the bases, for like 2 of the bases, maybe say it was like G and C, they had to name them fully, and only 15% of people could give the full name for those bases, which I thought was quite surprising. But it seems like you guys are all in that 15%, which is good. Yeah, exactly. I'm not going to write them out now to save time, but yeah, you do need to know the full name, not just the letter. But yeah, seems like most of you know them already, which is great. Cool. And yeah, there's purines and pyrimidines. You don't actually need to know that for AQA. So if you're not sure what those words are, it's because it's not on your spec. Everyone else needs to know purines and pyrimidines. But everybody needs to know the way they bond. So they have complementary base pairing, and they only bond with a specific other one. And yeah, exactly, Amy. There's a difference in the number of bonds they form. Cool. So let's look a little bit more closely at a nucleotide. So we can look at how we form this phosphodiester bond specifically. So I'm just going to draw out a nucleotide, and we can look at how it bonds. So this is the phosphate group up here. I'm just checking my notes to make sure I get the phosphate group correct. Oh, and I've not done it correct. I've done a double bond here, and it should be a single bond. At the moment, annoyingly, I usually have an eraser, but it's not working on my pen, so I can't rub anything out. I'm not sure why it's not working. But yeah, just pretend that bond's not there. So that's one nucleotide, and I'll draw the second one that it's going to bond to. So again, we start with the phosphate group up here. Got an OH, a double bond to an oxygen, and then it goes onto our ribose sugar. So this could be DNA or RNA. It would look the same for this bit. So hopefully what some of you might be spotting is how we're going to form this bond. It's much like forming a bond between a triglyceride, or between a fatty acid and a glycerol and forming a triglyceride, or between forming a polysaccharide. So yes, it's a condensation reaction. We can circle around a water that's going to come out. The oxygen could come from either side. It's completely random. It doesn't matter which one you draw it around. So you could just as easily draw around the other hydroxyl group on the bottom one and the hydrogen from the top group. It really doesn't make a difference. And then what you end up with is this oxygen joins the two together like that. So it's like an oxygen bridge between them. So yeah. So if we say these are the nucleotides, this is our polynucleotide. Going this way would be condensation. And going this way would be our hydrolysis reaction. Everybody happy with that? In fact, we have this page to check. So yeah, just give me a 1, 2, 3 to let me know how you are feeling with this. Or, of course, please feel free to use the emojis instead. I actually don't know how you would do that. Oh, there's a little button to click emojis. Okay, I see. Yes. Welcome to use emojis as well. In fact, I kind of prefer the emojis. Okay, some people on 3s, which is a little bit of a shame. Don't like to see that. Is there a specific thing that you're not sure about? A specific question? If you've got something, please feel free to ask now. It's better to get this out of the way with before we move on. If you're not following on. You don't need. Yeah, there's no ATP needed. Shouldn't the bond between the base and the pentase be in the corners? What do you mean? So the bond goes between the carbon 5 and the carbon 3. So if we go back, this is carbon 3 in the ribose. It goes 1, 2, 3, 4, and there's a 5th carbon that sticks up off here. So this is the carbon. This would be bonded to the carbon 5 on this one and the carbon 3 on this one. So this is what gives us the 5' to 3', or like vice versa, 5' to 3' structure of the DNA. So going this way, this is the 5' end, this is the 3' end. Any other questions? You're penalized for not drawing bonds on the corners. I'm not quite sure what you mean. Like on the, you mean how the way I've drawn ribose, or, yeah, I'm not exactly following. The corner of the base rectangle. Oh, okay. No, because that's not actually the structure. So I would say it's totally fine to draw it to the middle of that. That's not meant to be the structure of a base. This is not in. This is called like the skeletal form. So this part is in the skeletal form. This is actually representing what it looks like. This is just an oversimplification. So it doesn't really matter where that's going to. Yeah. So I didn't understand what you were talking about at first, but now I got it. Can you explain the 3' to 5' stuff? Yes, certainly. So this is basically how we orientate ourselves with which way the DNA is pointing. So we talk about 5' to 3'. This is referring to the carbon molecule. So in a nucleotide, we've got carbon 1, carbon 2, carbon 3, carbon 4. Carbon 5 is like sticking up out of the ring. This one up here is actually an oxygen. It's not a carbon. So the carbon 5 is here. So if this is the phosphate is on this end, it's bonded to the carbon 5. So the very top of this strand is the fifth carbon of the ribose sugar. So we call that the 5' end. And on this is only a polynucleotide consisting of 2 nucleotides. It's a very short one. But imagine it was a longer chain, and this is the bottom. This is the carbon 3. So the bottom of this DNA strand is carbon 3. We call that the 3' end. And one thing to remember is that DNA runs anti-parallel. So if one strand's going 5' to 3', the opposite strand, the complementary strand, must go 3' to 5'. So it will be flipped. The sugar phosphate backbone will be flipped in relation to the other one. Any other questions? What is a nucleic acid? How does DNA differ from RNA? We're going to cover that later. So hold that thought. Do hydrogen bonds have relevance to this topic? Yes, very much so. Hydrogen bonding is crucial for holding the DNA strands together. So they don't have such a significance in RNA, but in DNA, they're very, very important. Yes. Polypeptide bonds? No. Peptide bonds form between proteins. So they form a polypeptide chain, and that's made up of peptide bonding. But these are phosphodiester bonds in DNA formation. You do not need to be able to draw each base, no. And you don't even need to be able to draw the structure of the sugar other than being able to draw a pentagon. So that's all you need to know. So yeah, no, you're not expected to draw the structure of the bases. I don't think I know the structure of the bases, so it's like, it doesn't come up at all. Yes, in chemistry, hydrogen bonds are the strongest. Is that? I thought, yeah, well, they're definitely like the weakest bonds in biology, but I would have thought that would be the same in chemistry because it's all the same. It's all the same type of bonding. Okay, let's look at some exam questions. So again, the way we structure these classes normally is we do a bit of content, go over a bit of like revision, and then we'll do some exam questions aimed at what we've just learned. So the way to practice and to check. So this one, just a quite simple short answer questions here. First one is name the parts of the molecule represented by the letters Y. Oh, and X, I think it's supposed to say, because otherwise it's not worth 2 marks. So, yeah, Y is a nucleotide. Very good. And X is a base. Yeah, you can say organic base, nitrogenous base. Any of those is a good answer. Wombat, I would be careful about saying a specific one. Oh, sorry, no, actually you can. Oh, no, you're very correct. You could say that. I didn't notice there was a G here. So yeah, you could definitely say cytosine as well. Base / cytosine. Perfect. So next one, just a 1 marker. Which nucleotide base are common to DNA and RNA? A, C, and G. And you can write them in the short form to get full marks, or you can write them. You can write the full answer, the full proper name. Okay, so I can't remember who it was now who was asking about the similarities and differences, but here is a chance to have a go at them. So this question is for 3 marks. State 3 differences between the structure of DNA and the structure of RNA. So we're only looking at the structure, so don't give me anything about functions or anything like that. And we're looking for 3 differences. So you've got to be contrasting for these differences. Yes, probably the most obvious one is that DNA is double-stranded, whereas RNA is single, ready to mingle. or just single-stranded. Yeah, nice. What else? What else can you guys give me? Yeah, substitute the bases. Very good. Uracil in RNA and thymine in DNA. So it's really important whenever you're doing these questions that relate to giving a difference between things, or contrasting would be the same thing. Contrasting is like similarities and differences. Make sure you explain both of them. So don't just say there's uracil in RNA. That's not worth a mark. You have to say there's uracil in RNA and thymine in DNA. Otherwise, you're just giving a statement. You're not actually giving a difference. If you word it like only RNA has uracil, maybe that would be it's a bit better, but just give both sides. Yeah, probably the next most obvious one would be the type of sugar. So ribose in RNA and deoxyribose in DNA. And yeah, that's probably the three most obvious ones. Other things, I think I saw some people talked about the length. So DNA is long, RNA is shorter. But RNA can be clover shaped if it's tRNA, but it's not always. So I wouldn't go with that one. Oh yeah, a lot of you have clocked on there. DNA is linear. It can be linear, but not always. So DNA in eukaryotes is linear. Equals linear. But in things like prokaryotes, so bacteria, their DNA is actually circular. So it doesn't have a start and an end point. It's a circle. So I wouldn't go with that one. Yeah. Can we go through the genetic code? We're not gonna, this lesson is just about the structure of DNA and replication of DNA. We won't have time to go through protein synthesis in this one. That's like another lesson because it's quite a big topic. So we need to split it up. So let's look at the complementary base pairing now. So we're gonna look at the bases in a little bit more detail. So complementary base pairing occurs between which molecules, or sorry, which bases, I should say. A and T, G and C. So that's in DNA. Or A and U, G and C in RNA. Does anybody have a good way to remember that? Because I've never heard of a very good one, like a mnemonic or something to remember it. Or if you guys all just learnt it. I'll be curious if anyone's got a good one. Apple tree, car, garage. Apples on the trees, cars in the garage. Oh, that's like kind of the same thing, yeah. At the golf club. That's quite a good one, actually. Aquatango chewing gum. What's the Aquatango bit? I quite like this. At the golf club is good. Adidas trainer, Galaxy chocolate. Yeah, nice. At the group chat. Yeah, good. These are actually all very good. Using things like mnemonics is a great way to learn stuff in biology. Like probably the hardest thing with biology is just the broadness of the syllabus. It's a huge spec for any of the examples that you need to learn. So anything that helps you memorize it is a good thing. Usually, like the sillier, the better, because it sticks in your brain a little bit more. So yeah, no, it's good to see there's so many out there. Cool. So the way these bond. The line's already drawn on there from last time of teaching this class. A and T form 2 hydrogen bonds. Whereas G and C form 3. So there's bonding here and here as well. So we won't go into how hydrogen bonds form, but because it's like another thing, but people might recognize it's between delta positive and delta negative. So you might be familiar with delta negative and delta positive on hydrogens and oxygens in water, but you also get it on nitrogens. So nitrogens are slightly delta negative. Hydrogens, when they're bonded to nitrogen, just like when hydrogens are bonded to water, are slightly delta positive because both nitrogen and oxygen are stronger. They're better at pulling the electrons to them than hydrogen. So they sort of like slightly hog the electron. Purines and pyrimidines, yes, good. Thanks for reminding me. So yeah, if you're on AQA, you don't need to know this, but everybody else you do. So you've always got one purine bonded to one pyrimidine. So in this case, A and G are the purines. Sorry, not in this case. They always are. A and G are purines, and T and C are pyrimidines. The way to remember that is that the two words for thymine and cytosine, they're pyrimidines, which has got a Y in it, and so does thymine. And sorry, I've spelled that wrong. It's very difficult to, well, at least I find it difficult to talk and write. I end up just like confusing myself and writing what I'm saying at the time. Obviously, I'm not very good at multitasking. So yes, these are the pyrimidines, which both have the letter Y in, and that's a trick I used when I was learning to memorize it. Okay, what next? Yeah, let's look at some of the things that make DNA good at its job. So it's got a double helix. That makes it very stable. So the bases are bonded to each other, so it decreases the chance of mutations. Stable double helix structure protects bases, prevents mutations, or not prevents, but minimizes. So DNA was actually, we had RNA for a long time before we had DNA. RNA was the first type of nucleic acid that evolved. This is like billions of years ago. And we had RNA for quite a long time. And then DNA was it evolved from RNA, and it had certain advantages. And these are some of the advantages we're going through. So it was more stable. You're able to separate the strands. This is really useful for DNA replication. So when you separate them, both strands are able to act as a template, and therefore you can copy the DNA very accurately and very quickly. So this is a useful feature. DNA rep. Oops. What's the word we use to describe DNA replication? Oh, and yeah, actually that's a good thing. Let's do the enzymes as well. So DNA replication is semi-conservative, very good. And the enzymes that do it are helicase is the one that unzips it, aka it breaks the hydrogen bonds between the base pairs. And then, has everyone got it? Yeah, nice, DNA polymerase. DNA polymerase forms the phosphodiester bonds. It joins them together. DNA ligase, this is actually only in OCR and EdXL. You need to know about ligase. This is to stick together the Okazaki fragments. Yeah, exactly, on the lagging strand. If you do AQA, you might not have heard of that one because you don't need it. Nice. What is it for AQA? You just need these two. DNA polymerase and helicase. You don't need to know about ligase, which is lucky. the reason it's synthesized from that direction is because DNA polymerase can only bind to the 5' end of the ribose, so it has to go in that direction for replication. Yeah. AQA is easier than OCR. Possibly. There's definitely less content in AQA than OCR. OCR is a very big spec. It is a little bit hard. But then the trade-off is because it's so big, the grade boundaries tend to be lower for OCR. So something like, I think one year, if you got I think it was as low as like 67% or something was an A star. So it was like a very low. They realized that the test was very hard because obviously in a test, you're not just trying to beat the test. It's all comparative. It's all relative. So if a lot of people do well, they'll push the grade boundaries up or down to match that. So you basically need to be beating everyone else. If you want to be an A star, you need to be in the top 10% of people who have done that exam board. So it doesn't really matter how hard it is. You just need to be in the percentile that you want to be getting. So if you want to get an A, you need to be in the top, or an A or an A star, you need to be in the top 25% of people who sit that paper. So it's actually more important who's sitting your paper than which paper you're doing. Oh, sorry, we didn't finish this last one. So it's a large molecule, so it carries lots of information. A field of biology we're like looking at getting into is actually storing information in DNA because it's a very good way to store info. So like kind of like having a biological hard drive where we can store information, actually like putting stuff in the DNA, which would be quite cool if they can, well, they can already do it rudimentary, but get this more up and running. Okay, how's everyone feeling with that? Give me an emoji or a marks score out of 1, 2, or 3. Perfect. So we've got just under 15 minutes left. We're going to do one more set of past paper questions to check what we've gone on. So check what we've done, and then I'll just talk you through the website quickly for the last few minutes. The enzyme primase is for another lesson. It's like how the DNA, the actual, this is a protein synthesis, how they can actually attach the RNA polymerase. But we're not going to talk about it today. And again, it's gone now, but someone asked about Okazaki fragments. Again, we're not going to have time for that today, unfortunately. But if you do go on our website, there's a nice video explaining all of that. Can we do semiconductor replication? Again, I'm afraid we don't have time for all of that today. We just got these last set of questions. But yeah, I would love to talk you guys through that another time. If you do subscribe, we've got drop-in sessions and web classes, and we go through all that stuff. Yeah, this will be available on YouTube, this class, exactly. So final questions. First thing, we would need to name the base replaced by letter T. Oh, sorry. I literally just read that wrong. Not replaced, represented by letter T, so thymine. Next one for T marks, draw on the complementary strand to this DNA molecule. Yes. Nice. And Marion, thanks for the support. I recognize you from your name from the classes. Is this Amy Scott? Are you the Amy that normally comes to classes as well, or a different Amy? Yes. For those people who've got the answer, A, C, G, C, G, U, and A. We would form. Cool. So final question. Let's look at this. This is quite a common style question that comes up in all the specs. You'll be given some of the percentages of the DNA, and you have to fill in the rest from that. So it's basically just testing your knowledge of complementary base pairing, and then being able to subtract a number from 100. Can you hear the bottle squeak? Sorry, yeah, it's so distracting. It just like, it's, yeah. I'll stop drinking out of it. I can go 10 minutes without a sip of water. Cool. So if A on strand 1 has 16% of the bases, so these are all in percent, so we know it adds up to 100. Therefore, the T of this one must have 16%. If the C of this one has 21, the C of this one has 21, and if this one's got 34 for G, this one must have 34 for C. Then to work out the remaining one, so it's A or T, we just take these numbers, 16 plus 34 plus 21 equals 71, I hope. We then, because it's percentage, we do 100% minus 71 equals 29. And that's our answer. 29, or not our answer, that's how we fill in the last columns on that thing. Die of dehydration. I'll be fine. Thank you for your concern though. Why are the percentages on the opposite strands the same? Because of complementary base pairing. So when DNA forms, the bases will only bond to a specific ones. These two will only bond, and these two will only bond. So every time you've got an A on one strand, you will have a T on the other strand. That's why they're always exactly the same. There could be a mistake in the DNA, but this is very rare. So DNA is very good at copying itself. So the rate of mutations coming in is extremely low. I think there's a comparison to how often like a human would make a mistake when they're just copying out numbers, and it's like millions of times better than we are at doing it, which is lucky because it happens a lot in our cells. Aren't the percentages for A and T meant to be the same? Only on opposite strands. Otherwise, yeah. Mistake in the first question. Where? The first part. What's the mistake? A U instead of a T. Oh no, because this is because this is a strand of messenger RNA. But no, thanks for drawing this up, because I probably should have made this clearer at the time. So this isn't a complementary DNA strand. I can see why this is confusing, because we haven't really been talking about protein synthesis in this lesson. We've been talking more about DNA. So this is actually a messenger RNA strand. And messenger RNA is, well, obviously made up of RNA, which uses uracil instead. But yeah, no, good to point that out, because I should have made that clearer as we were going through. Yeah, cool. Hopefully that clears that up. I was slightly panicked. Whenever someone says there's a mistake, I'm always like, oh no. But that one was easy. Okay, perfect. So that was like a sample of what a class would be like. I now just want to talk you through our website. So I'm just going to close this PowerPoint. We'll just spend 5 minutes looking at the structure of our site. So what do I want to do? Close this. Open the site. So this is our beautiful website. It's looking very swish. This is like the home page. What you would do if you wanted to join is we've got some different packages. So 3 levels of package. The basic one is access to the videos and exam walkthroughs and mini revision guides. I'll sort of talk you through those in a minute. this is the middle package. It's got a few more things, these self-marking quizzes, and the thing actually tracks how you're learning and sort of goes through that. And then the ultimate package, this is the most deluxe offering. It's got all the same things as the other ones, but it's also got these web classes and drop-in tutors and chat support. So those are available for the premium packages. So the web classes are pretty much like what we've done today, but it will be with a much smaller group. So there's usually between like 5 and, I'd say like on average, like 5 and 10, sometimes it's up to 15. But yeah, generally like much smaller classes. So yeah, I think it's definitely worth the money. If you compare it to tutoring, which would be around £50 an hour for a tutor, at least in London, that's the going rate. You can do, so for each subject, we've got 4 web classes and 4 drop-ins a month. So that's 8 tutoring sessions effectively. And yeah, you can also get this tutor support chat. So we've got loads of great videos which go through the content. If you've watched the video and you're still not sure what is going on, you can just drop a comment on the video and a biology teacher will answer that question for you. Perfect. Any questions? Yes, you're very welcome. I will show you the actual format of what this looks like. So this is like the AQA course. I'll go on AQA because it's probably the most common exam board, or like the most people are doing it. We'll look at the ones we were doing today. So we went through nucleic acids today. So we scroll to that. For each topic, you start with a diagnostic quiz. What this does is it tells you which parts of that topic you know well and which parts you need a bit more practice on. So it's really useful because you've only got so many hours in the day and so much time you can devote to revision. You've got to revise smart as well as hard. So if you can target your revision to where it's most needed, and this information is provided by these diagnostic quizzes, you can get more out of your revision session. So this is like the diagnostic quiz. It's 10 questions. So I'm just going to, as a way to skip through them. I don't want to go through all 10, but you see the structure. You basically answer these 10 questions. That will give you a basis for how well you know that topic already. So you know how much time you need to devote to learning it. You then get a section like this. You've got one of these for each topic. So this is the nucleotides one. You've got videos that talk you through the whole process. So they're really clear, logical. So you should be pretty comfortable with it after watching those videos. If you're not, and you're on the ultimate package, this is where you can ask questions and people will answer them. So this is one that, I've answered this one. Yeah, so if you're still not sure, ask a question. One of us will answer your question for you. So that's a really useful feature. You've then got some quizzes again to check your understanding. And again, these will mark and refer back. So if you get a question wrong on a quiz, not only will it tell you you got the question wrong, it will then link you back to the relevant video to talk you through the process. Is there a free trial like LeHub? I haven't heard of LeHub, but yes, we do have a free trial. You get 1 week free trial. So yeah, please feel free to try that out. Is there Edexcel International? Yeah, unfortunately, we don't have the international A-levels yet. We're working on those and hopefully we'll have them soon. But we do have the normal Edexcel board, which is like fairly similar. There are some differences. So if you did that, you would have to occasionally refer to your own syllabus, your spec points, just to make sure they are matching up. Does subscription include A-level maths? You need a subscription for each subject. So we do have a maths one. We do have maths. Yeah. When will we have international A-level? If you put that question to, what is the email? I think it's support@SnapRevise.co.uk and ask there. Yeah, that's a good question. And I think seems to be like a reasonable amount of demand for it. So hopefully we will have a package specifically targeting those already. Oh, sorry, this is the email, SnapRevise.co.uk. So questions like that, when will we have this? These packages, ask them there. We have live streams like this for the other subjects. Yes, we have one a week for alternating between the different science subjects. So I'm just scrolling back to see if there's any other questions that you guys have. Do you need it for English and maths? We do have some GCSE packages, and we are working on making them. At the moment, they're not quite as detailed as the A-level packages. For example, there's no web classes or drop-in sessions, but that might be something that will come in the future. Cool. So let me go back to the PowerPoint to share with you the promo code. So, I mean, you can probably see it already. Switch. Yes, so our code for today is DNA 10. So that gets you 10. Oh, sorry, I thought it was 10%. It's actually better. £10 off your first month. So 20. If you're doing the ultimate package, that's 20% off. If you're doing the basic package, that's a free month. So that's pretty good. Will there be a physics cheat sheet? We do have like very summarized, condensed like notes for each topic, which basically are cheat sheets. Yes, exactly. When are we announcing the winners of the competition? That will be tomorrow. So you have to enter it today. So tag SnapRevise in your Instagram and share a post. And then tomorrow the winner will be picked at some point. I don't know exactly which time, what time, but at some point tomorrow. English A-level? No, we don't have English yet. Again, hopefully coming in the future. I think the ones being worked on at the moment are, what were they? Psychology and economics, I think, are the next two, but hopefully eventually there'll be all the A-levels, exactly. Yeah. Is the story mentioned fine? I actually don't know that much about Instagram, and I'm not 100% whether you have to tag it, like upload a picture or story mention. If you're not sure, you can email that, email hello@snaprevise.co.uk. But I'm pretty sure either of those would be fine. Yeah. Perfect. Cool. So unless you've got any questions, that's the end. I'll hang around for a few more minutes. If you've got questions, please feel free to ask. But otherwise, thanks for joining. Hopefully, you found it a useful session and you learned something about biology. And please subscribe to our YouTube channel. We've got these classes once a month, so set a reminder so that you remember to join. And I hope to see some of you guys in the web classes on the website in the next few weeks, hopefully. Yes, see you tomorrow, Marion. You're very welcome. Have a good evening. Bye-bye. You're welcome. You're welcome. Salim, yes, what is your question? Fire away. I'm going to hang around for a few minutes. Do I have a Discord server? I'm afraid I don't understand the question. What is that? That's probably one to send to hello@SnapRevise, because I don't think I'm the right one to answer that. I'm not sure what that is. Alejandra, I've never seen Alexandra in the Spanish way before. Alejandra, or is it Spanish or is it Portuguese? Sorry, anyway, your question. If there's an exam question you don't understand, can the tutors walk you through? Yes, definitely. So that's on the drop-in sessions. You need the ultimate package for that. You can either ask a comment under the video and then someone will reply to it, one of the tutors, or you can send that question in and we can go through it on like a live talk, like talking through the answer in a drop-in session. So there's two options for that. Discord is a social media thing. Okay. Oh, I'm too out of the loop these days. I'm aware of TikTok, and that is the most recent one that I know. Spanish. Okay. Nice. Yeah, you're very welcome. Shrek for life. Love the name, by the way. Yeah, you can rewatch this anytime. It will be uploaded onto YouTube after the session. It might even be instant. I'm not sure exactly when it'll go up. But yeah, you can definitely rewatch. And you can also scroll back through our old videos as well. We've got lots of them on YouTube. Cool. Any more questions? Or yes, Salim? What's up? Yes. If you do the ultimate package, you can look back through all the historic web classes. Yeah, so even if you join now and we've gone through pretty much most of the first term's worth of content, you can obviously backdate and go back through all those old lessons. So yeah, you will have access to all of that. When's the next biology A-level stream? I think it might be in the new year. So we've done biology today, and then there'll be the other subjects. So probably not till 2021, which seems weird. 2020 has flown by, and it's been the worst year. Just, well, not the worst, but I don't know. I don't think anyone's loved 2020. Let's hope 2021 is a bit better. So yeah, I think probably next year, early Jan for the next class. Any other questions, or are you guys all good? Perfect. Okay, thanks for joining. Oh, one more. Just A-level and some GCSE ones as well. So we're just doing the A-level, the UK courses at the moment. Maybe in the future branching into IB or some of the other country exam boards. But yeah, for the moment it's just UK stuff. What will the topic be? I haven't decided yet. To be arranged. Probably an A2 topic though, year 13 stuff, because we did year 12 this time. Does this get saved? Yes, it'll be uploaded. Yeah. Cool, right. Have a great evening, guys. Thanks for joining. Hope to see you in the future, either on YouTube or on SnapRevise. Cool. Thanks. Bye-bye.